Now length of DNA in a diploid cell, it comes out to be 2.2 meter. What is the dimension of nucleus? We know that DNA is present inside the nucleus. Dimension of nucleus, that is the space or the diameter, it is approximately, it is equal to 10 raised to power minus 6 meter, एंड द लेंथ ऑफ डीएनए इज 2.2 मीटर. तो ये इतनी लेंथ का डीएनए इतने डायमेंशन में कैसे अपने आप को एडजस्ट करेगा? लेट अस सी. दिस इज डन इन द टॉपिक पैकेजिंग ऑफ डीएनए हेलिक्स. इन केस ऑफ prokaryotes, we know that in prokaryotes there is no nuclear membrane and the nuclear material lies in contact with the cytoplasm. There is a single circular DNA which remains attached to the cell membrane at a point, and this single circular DNA is called nucleoid. This is how DNA is packed in prokaryotes. But in case of eukaryotes, the cells, they have a nucleus, and inside the nucleus the DNA is present. How it is present? Let us see. Packaging. Now, DNA is negatively charged and it is associated with some proteins called histones. In न्यूक्लिक एसिड डीएनए और हिस्टोन की जो रेशो है दैट इज 1:1. 50-50% दोनों प्रेजेंट है. डीएनए इज नेगेटिवली चार्ज एंड डीएनए इज एसोसिएटेड विद हिस्टोन प्रोटीन्स एंड मेनली two histone proteins present are lysine and arginine, which carry the positive charge. So कैसे ये पैक्ड है? Histone is present in the form of a octamer. In this octamer, this is a histone molecule, and each histone molecule has two core particles, और दोनों core particles they are made up of 4 proteins. कौन कौन से हैं वो चार? four different histone proteins. They are H2A, H2B, they are H3 and H4. Same H2A, H2B, H3 and H4. One histone molecule is called octamer because it consists of two core particles each having 4 different proteins. तो चार protein हो गए 8. So this is octamer, and there is one protein which is called, this is the H1. So this is the protein histone and it is wrapped around by DNA. Then it goes like this, another histone, and DNA is wrapped like this, and this is histone and this. तो कैसा हमको स्ट्रक्चर बनेगा? ये histone wrapped by DNA. This particle is called nucleosome. Then nucleosome. ऐसे DNA इस पे coiled था, then ऐसे, then coiling, then straight coiling and coiling. This is how DNA is wrapped. These are the nucleosomes, and the portion of DNA between two nucleosomes, this is called DNA linker. So a nucleosome plus a DNA linker, यानी इतना portion. A nucleosome plus a DNA linker, it is called chromatosome. This is chromatosom. So many and this appears like beads on a string, and it forms thin chromatin fibers. So let us see the histone particles. Histone particle, it is a octamer which forms a core. Core has two molecules, and two molecules has four different histones: H2A, H2B, H3 and H4, and there is a fifth histone H1 which attaches the end. इंटरफेस स्टेज में जो है ये कैसे प्रेजेंट है? दीज आर द न्यूक्लियोसोम, डीएनए लिंकर्स, एंड वन न्यूक्लियोसोम एंड डीएनए लिंकर, इट फॉर्म्स अ chromatosome, and it appears as beads on the string. And during packaging, another protein, non-histone chromosomal protein, is also present. A nucleus shows two regions, euchromatin and heterochromatin. Euchromatin is the portion which is loosely packed, where the DNA is loosely packed. It stains light, and transcriptionally active means this portion undergoes transcription. It helps in the formation of messenger RNA. Heterochromatin, it is densely packed and stains dark, and this is inactive transcriptionally. That's all for today.